A pleasant day to you, icons and lady icons. Welcome to the ninth module of our discussion in the subject General Biology 1, in which we are going to talk about the concept of the second half of bioenergetics, which is all about the cellular respiration. Now, whenever we're actually talking about cellular respiration, this is not the same as the concept of breathing. Because whenever we are considering the concept of breathing, it is just the inhale and exhale of the different gases that is present in our body. Whereas in the case of cellular respiration, it is not only about the presence of the oxygen gas, but rather it is also the interaction of the oxygen gas with the glucose molecule that we consumed and how we actually exhale carbon dioxide in that certain type of process. Okay, so since we are talking about the second half of bioenergetics, let's have a short review regarding the concept of photosynthesis, since this is the very first part of bioenergetics that we discussed last module. Now, remember that in the case of photosynthesis, some of the energy in sunlight is captured by the chloroplast using the pigment known as the chlorophyll. Then, atoms of carbon dioxide and water are rearranged and glucose and oxygen are produced. And in the case of the cellular respiration, it is now the time in which we are going to break down glucose molecule to carbon dioxide and water, and at the same time the cell captures some of the released energy to make adenosine triphosphate, or the energy currency that we are using in our body. And remember, cellular respiration takes place in the mitochondria of eukaryotic cells, or simply known as the powerhouse of the cells. Now, in the case of bioenergetics, specifically in the cellular respiration, remember that this is the second half of the concept of metabolism. In this case, it is now the catabolism phase in which we are going to break down glucose molecule, but we're still going to create other substances that will be useful to other organisms. For us humans, we are actually going to utilize the adenosine triphosphate that will be created in this process. If during the discussion regarding the Calvin cycle, we simply focus on the different components and the different materials that were created during that certain type of cycle, in cellular respiration, we're actually going to focus on the different materials that will simply be utilized and that will simply be created during the process of the cellular respiration, or also known as the aerobic respiration. In the case of us, eukaryotic organisms, since we are also associating the concept of cellular respiration with breathing, it actually involves the presence of oxygen. And if cellular respiration involving oxygen is considered, it is known as aerobic respiration. But if there are no oxygen that is present in cellular respiration, it is now known as anaerobic cellular respiration. Anaerobic cellular respiration is happening both on prokaryotic and eukaryotic organisms. But later on, we're actually going to discuss what type of cells, what type of organisms are actually undergoing these different types of cellular respiration. In the case of photosynthesis and cellular respiration, always remember that it is just like the utilization of energy that is first coming from the sun, and it will be captured by the photosynthetic organisms. Then it will create glucose and oxygen. Then the oxygen will simply be inhaled by, for instance, us human beings, and the glucose will simply be consumed by the consumers, in which the cellular respiration will now happen in the mitochondria, and adenosine triphosphate will be released and utilized at the same time in the presence of heat energy, or basically the presence of the different types of energy that will simply be utilized by our body. Then, the presence of the carbon dioxide and oxygen will actually be the product of the cellular respiration that will now simply be fed on the process of photosynthesis, just like what we had discussed on module number 8. As you can observe, photosynthesis and cellular respiration go side by side. But please take note, the discussions that we're actually having in this General Biology 1 class is just scratching the surface of all the biochemical pathways that are present on our body. This is actually the most simple of the different types of biochemical processes. There are different types of biochemical process that is happening on the different organs in our body. Since it was mentioned that photosynthesis and cellular respiration go side by side, let's now consider it in a very scientific manner. Now, if we can still recall, on our discussion when it comes to photosynthesis, we discussed here the presence of the redox reaction in which carbon dioxide will be reduced to create now the glucose, while water molecule will now be oxidized to release oxygen gas. In the case of cellular respiration, as you can observe on the lower portion of our screen, glucose will now lose hydrogen atoms and becomes oxidized. Remember, just like what happened to water, it lost its hydrogen bonds. While in the case of glucose, in the case of cellular respiration, glucose molecules will now be oxidized to release carbon dioxide, while oxygen gas will gain hydrogen atoms and becomes reduced. During this process, adenosine triphosphate and heat will simply be released. Okay, so please take note, when it comes to the relationship that is present when it comes to photosynthesis and cellular respiration, they are just the vice versa of each other. When we are talking about the aerobic respiration, there are actually three main phases that we are going to discuss in this video discussion. The first phase, we have the presence of glycolysis. The second phase, it is now known as the pyruvate oxidation and citric acid cycle. The third phase, it is now known as the oxidative phosphorylation, in which we are going to cut this video discussion based on those different types of phases. For this part of the video presentation, let's start first with phase 1, which is the glycolysis. Now, in the case of glycolysis, it is actually happening on the cytoplasm of eukaryotic cells. And in the case of the pyruvate oxidation, citric acid cycle, and at the same time the oxidative phosphorylation, or also known as the electron transport chain or chemiosmosis, it is now happening inside the mitochondrion or the powerhouse of the cell. Let's start with glycolysis. Remember that the phase 1 occurs in the cytoplasm and begins cellular respiration and breaks down glucose into two molecules of a 3-carbon compound known as pyruvate. We're going to start with a 6-carbon glucose molecule, but we're going to cut it to a 3-carbon compound known as pyruvate. Then, in the process of glycolysis, we are actually considering here the presence of two major stages. The first stage, it is the energy investment stage, while the second stage, it is now known as the energy payoff stage. Because for the very first stage, we are going to utilize the adenosine triphosphate that were simply created on the previous cellular respiration cycles. Then, on the second stage, it is now the energy payoff, in which we are now going to utilize and create adenosine triphosphate that would be needed by that organism. So, glycolysis in summary, a single molecule of glucose is enzymatically cut in half through a series of steps. Two molecules of pyruvate are produced, two molecules of NAD+ are reduced to two molecules of NADH, and a net of two molecules of ATP is produced. Please take note, in the case of cellular respiration, we are now utilizing here NADH and charge NAD. That in the case of cellular respiration, we are utilizing here nicotinamide adenine dinucleotide. We are not utilizing the same substance with phosphate. Now let's start with the very first stage, known as the energy investment stage. From the term itself, it is known as energy investment because we are going to utilize adenosine triphosphate to help the cutting of the glucose molecule to two pyruvate molecules. So during the very first stage, there are actually 5 basic steps. The very first one, it is now the presence of a glucose molecule, and with the aid of hexokinase, which is an enzyme, it will now attach the third phosphate that is present in adenosine triphosphate to create glucose 6-phosphate. Okay, why is it known as glucose 6-phosphate? Because it is now possessing carbon 1, 2, 3, 4, 5, 6. And this is now where the phosphate group will simply be attached. Okay, if we're actually going to observe when it comes to your module, and I am actually going to advise that while we are doing this discussion, you should take a look on your module for you to know what I am simply talking about. Okay, this discussion is not enough if you're not going to consider taking down notes or observing what is present on your modules. Okay, the very first step, it is now the transformation of glucose molecule to glucose 6-phosphate, and it is being accomplished by the enzyme known as hexokinase. Then, the second one, glucose 6-phosphate will now interact with the enzyme known as phosphoglucose isomerase, which will now create fructose 6-phosphate. From the name itself, it is known as phosphoglucose isomerase because it will now target a glucose molecule with a phosphate group, and isomerase, remember, whenever we're talking about isomers, they are the same chemical formula but different structure. Now, in this case, from glucose, it created fructose molecule. The reason why it created the fructose molecule is because of the attachment for step number 3. It would be easily for another phosphate to attach on a fructose than on a glucose. So on step number 3, with the presence of the enzyme known as phosphofructokinase, it will now attach the third phosphate that is present on adenosine triphosphate to the fructose 6-phosphate, creating now a product known as fructose 1,6-bisphosphate. And adenosine triphosphate would simply give off its third phosphate, creating now adenosine diphosphate. As you can observe, how many adenosine triphosphate have we utilized during the energy investment stage? We utilized 2. We invested 2 ATPs, okay, in which we are going to have a payoff on the second half of the glycolysis. Okay, so please take note, from fructose 6-phosphate, it interacted with phosphofructokinase to create now fructose 1,6-bisphosphate. Carbon 1, 2, 3, 4, 5, 6. And if you're actually going to observe here, these are the close models of glucose and fructose. But hopefully, if you're going to have a specialized discussion when it comes to your collegiate level biochemistry, you're going to understand why glucose and fructose look like this whenever they are considering the closed model, just like a piece of piatos. Okay, now for the last part of the energy investment stage, it is now the presence of the enzyme known as aldolase. The enzyme aldolase will actually split fructose 1,6-bisphosphate to 3 carbon molecules. With the presence of aldolase, it will actually create now a G3P or glyceraldehyde 3-phosphate, which is present on the first half of the fructose 1,6-bisphosphate. Then the second half, it will now create a dihydroxyacetone phosphate. Now, with the presence of the enzyme known as isomerase, it will now transform dihydroacetone phosphate to glyceraldehyde 3-phosphate. Since glyceraldehyde 3-phosphate is the easier type of molecule to be utilized on the second half of glycolysis, dihydroxyacetone phosphate will simply be transformed to glyceraldehyde 3-phosphate, so that when we enter the second stage, we are now going to utilize 2 molecules of glyceraldehyde 3-phosphate and not 1 glyceraldehyde 3-phosphate and 1 dihydroxyacetone phosphate. Please take note that the enzyme isomerase will create glyceraldehyde 3-phosphate from dihydroxyacetone phosphate. From the energy investment, let's now consider the energy payoff stage, in which we are now going to create here adenosine triphosphate. So what will simply happen? Now, here's the thing. Since we are going to start the step number 6, or the second stage, which is known as the energy payoff stage, since we are going to start the second stage of glycolysis with 2 molecules of glyceraldehyde 3-phosphate, the enzyme known as glyceraldehyde 3-phosphate dehydrogenase will now actually remove hydrogen ions from these compounds, And while the hydrogen ions are released, it will be accepted by the NAD plus to create now charged NAD, which are known as the NADH. While that is happening, phosphate group will actually be attached on carbon number 1, creating now a 1,3-bisphosphoglycerate or BPG or BPGA, just like what we utilize during the process of photosynthesis. Okay, so once again, on step number 6, glyceraldehyde 3-phosphate dehydrogenase will interact with G3P. Then, hydrogen ions will be released on carbon number 1, and on carbon number 1, the phosphate that is present on the cytoplasm will simply be attached on carbon number 1 to create a new compound known as 1,3-bisphosphoglycerate or BPG. Now, on the 7th stage, since there is now the presence of extra phosphate on carbon number 1, the phosphoglycerate kinase will now actually detach that phosphate that is present on carbon number 1 and will be attached on the adenosine diphosphate, creating now adenosine triphosphate. Since there are two 1,3-bisphosphoglycerate that was simply created on the second half of glycolysis, 2 adenosine triphosphate will be released on 7th step. Okay? On the 7th step, 2 adenosine triphosphate will be released since there are already 2 molecules of 1,3-bisphosphoglycerate. As simple as that. Then, it will now create a substance known as 3-phosphoglycerate or the 3PG. If we're going to observe the structure of 3-phosphoglycerate, it is not the same as the structure of the glyceraldehyde 3-phosphate, since it is not actually possessing aldehyde functional group, but rather it is just a glycerol backbone with a double bond of oxygen and single bond of oxygen on carbon number 1. It is no longer aldehyde. Okay? Now, in the case of the 3-phosphoglycerate, it will now actually undergo another transformation in which the enzyme known as phosphoglyceromutase will now transform the 3-phosphoglycerate and move the phosphate group from carbon number 3 and move it to carbon number 2. Okay? The enzyme known as phosphoglyceromutase, from the name itself, it is now possessing mutation. It will now mutate the 3-phosphoglycerate. Then, the phosphate group that is present on carbon number 3 will now be moved to carbon number 2, creating now another compound known as 2-phosphoglycerate. Okay, that is just the enzyme that is simply utilized for that process. Then for step number 9 is now the enzyme enolase. Enzyme enolase will now remove water molecule from carbon number 3 to create now phosphoenol pyruvate. And lastly, for step number 10, the phosphoenol pyruvate will now interact with the enzyme known as pyruvate kinase, and the phosphate that is present on carbon number 3 of phosphoenol pyruvate will now be released, will be attached to adenosine diphosphate, creating now adenosine triphosphate, and a new compound known as pyruvate will simply be released, which will be utilized on the next part of the cellular respiration. Okay, so please take note, since we are now considering here the presence of 2 stages, the energy investment stage and the energy payoff stage, which will now create pyruvate. 2 molecules of pyruvate will be created by 1 molecule of glucose. If you're considering this glycolysis as somewhat complicated, please take note that we are simply talking about only 1 unit of glucose. There are thousands, 10,000s, and even hundreds of thousands unit of glucose that is present on 1 grain of rice. Imagine how many times glycolysis is actually happening on our cytoplasm at this very moment while you are watching this video discussion. Now, for the summary, there are different stages that we are going to consider now, the different products that were simply be created. We have the glycolysis, in which we discussed a while ago. Then, how many NADH were simply created? It created 2 NADH. Okay? Then, how many ATP were simply created? We created 4 ATP. And lastly, for the last column, how many ATP were used? We utilized 2 ATP during the energy investment stage. Okay? 2 ATPs were utilized during the energy investment, while 4 were simply created during the energy payoff stage. Okay, so that's the thing whenever we are talking about the concept of glycolysis.